The goal of this video is to describe an integrated pipeline for the design, fabrication, and use of peptide microarrays for profiling the specificities of histone antibodies.
Antibodies that recognize histones and their diverse post-translational modifications are key reagents in chromatin biochemistry research. We rely on these tools to deepen our understanding of histone modification function. Recent studies reveal inconsistencies in histone antibody behavior. As antibody choice is a most critical element in epigenetics experiments, rigorous and comprehensive quality control measures are demanded for these reagents.
Here, we demonstrate an integrated pipeline we developed for designing, fabricating, and analyzing peptide microarrays to generate specificity profiles for antibodies that recognize histones and their post-translational modifications.
The pipeline is facilitated by ArrayNinja, an open-source interactive software application that we developed, which integrates the design and analysis stages of microarray experiments.
To begin designing the array slide, under the Plan a Slide Layout heading, select the microarray printer that will be used. Then, type Empty in the Load Plate field and press Enter. Set the spot diameter to 275 micrometers and the spot spacing to 100 micrometers. Fill in the remaining parameters appropriately for the experiment, ensuring that the layout allows for sufficient replicates. In the resulting layout preview, mouse over each unique peptide feature on the slide and enter a feature identifier in the pop-up text box. Once each unique feature has been labeled, click Populate. Name the slide layout.